children, welcome back. I am Dr. Jisha Joe. In this session, we shall discuss a few more important multiple choice questions from the chapter Photosynthesis. But before we move on with the discussion, let me just remind you to do subscribe to my channel, and if you find my videos helpful, do share with your friends so that they too can be benefited. Okay children, so let us now move on to question number 1 of part 2 of the MCQs from the chapter Photosynthesis. Which among the following is not needed for photosynthesis? Options are chlorophyll, water and carbon dioxide, glucose and oxygen, light energy. Question is, which among the following is not required for photosynthesis? What are the raw materials for photosynthesis? Just look at the options once again. Chlorophyll, water and carbon dioxide, glucose and oxygen, light energy. Did you get the answer? The correct answer is option C, glucose and oxygen. Glucose and oxygen are the products of photosynthesis. Okay. So, they are not needed for photosynthesis. They are actually the products of photosynthesis. Now, moving on to the next question. Which is correct with regard to sugar concentration theory? Stomatal opening and closing depends on the generation of a potassium ion concentration gradient. Okay. Second option, ATP is used to actively pump sugar from the adjacent cells into the guard cells. Option C, increased potassium ion concentration in the guard cells make them hypotonic, resulting in endosmosis. And option D, increased glucose concentration in the guard cells increase the osmotic pressure of the guard cells. Which is correct with regard to sugar concentration theory? So, we have two theories, the sugar concentration theory and the potassium ion concentration theory, when we study the mechanism of stomatal opening and closing. Okay. Now, according to the sugar concentration theory, what is actually happening? We know the guard cells have chloroplast in them. During daytime, what happens? Because of the presence of chloroplast inside the guard cells, photosynthesis will take place in the guard cells, and as a result, the sugar concentration inside the guard cells will increase because of photosynthesis taking place in the guard cells. During daytime, the sugar in the guard cells increases. There will be an increase in the glucose concentration or sugar concentration in the guard cells, making the guard cells hypertonic. Okay. So, what will happen? Endosmosis. Water enters into the guard cells from the adjacent cells. Endosmosis will take place and the guard cells become turgid. They swell and the stoma or the opening opens up, the stomata opens up. That is actually what is happening in the sugar concentration. According to the sugar concentration theory, this is how the stomata is opening. And when photosynthesis is not taking place, what will happen? Water withdraws from the guard cells. Water moves out of the guard cells into the adjacent cells, and the guard cells become flaccid and the stoma closes. Now, look at the options once again. Here the question is, which is correct with regard to sugar concentration theory? Option 1, option A, stomatal opening and closing depends on the generation of potassium ion concentration gradient. So, here there is no potassium ion concentration gradient, no development of such a gradient there. Okay, so that option is wrong. Second one, ATP is used to actively pump sugar from the adjacent cells into the guard cells. That is also wrong, because the sugar or the glucose is produced in the guard cells because of the presence of chloroplast in the guard cells. Okay, it is not pumped from the adjacent cells into the guard cells. The sugar or the glucose is produced as a result of photosynthesis in the guard cells, because the guard cells contain chloroplast in them. And option C, increased potassium ion concentration in the guard cells make them hypotonic, resulting in endosmosis. That is also a wrong statement. Here this is not related to potassium ion concentration theory, and that option is also wrong. Suppose an increased potassium ion concentration in the guard cells make them hypertonic, not hypotonic. Okay, so that is also a wrong option. And come to the fourth option, option D, increased glucose concentration in the guard cells increases the osmotic pressure of the guard cells. Yes, that is true. Because of the presence of more glucose in the guard cells, the osmotic pressure inside the guard cells increase, and as a result water enters into the guard cells from the adjacent cells. Endosmosis will take place into the guard cells, and the guard cells swell up or bulges out, and the stoma opens because of the peculiar type of thickening of the guard cells. The stoma or the stomata will open up. So the correct answer is option D. Increased sugar concentration in the guard cells increases the osmotic pressure of the guard cells. Yeah, the remaining is not being seen there. Okay. Okay, option D is the correct answer. Yeah. Increased glucose concentration in the guard cells increases the osmotic pressure of the guard cells. Okay. Now moving on to the next question. Number of water molecules required for the synthesis of one molecule of glucose. For the synthesis of one molecule of glucose, how many water molecules are required? Just think of that equation on of photosynthesis. You will know how many water molecules are required. Look at the options. Option A 6, option B 12, option C 1, option D 2. So just think of that equation to represent photosynthesis. 6 CO2 plus 12 H2O gives one molecule of glucose, C6H12O6. Okay, so how many water molecules are required? 12 water molecules are required for the synthesis of one molecule of glucose. How many carbon dioxide molecules are required? 6. 6 carbon dioxide plus 12 water results in the formation of one glucose molecule. Okay. So the correct answer is option B 12. Next question. Products of photolysis of water. What is meant by photolysis? Photolysis. Lysis means splitting. Photo, that is light. So splitting in presence of light. So water is split in presence of light here. So what is the product of photolysis of water? Water splits up into what all? Look at the options: H plus ions, electrons, oxygen, NADP, electrons and H plus ion, NADP and ATP, NADPH, ATP and oxygen. Which is the correct answer? Water splits in presence of light to form protons, electrons and oxygen. So the products of photolysis of water are option A, H plus ions, electrons and oxygen. That is the correct answer. Okay, now what happens to these products of photolysis? What happens to the H plus ion? The H plus ion combines with NADP plus in presence of electrons to form NADPH. Okay, nicotinamide adenine dinucleotide phosphate, reduced form of NADP is produced. So that is what is happening to this H plus ion, which is produced as a result of photolysis. What happens to the electrons? The electrons are used for the photophosphorylation of ADP in presence of phosphate, or ADP in presence of the electrons combined with phosphate group to form ATP, adenosine triphosphate is produced. And what happens to the oxygen? The oxygen is given out as molecular oxygen. So this is what is happening to the products of photolysis. What are the products of photolysis? The products of photolysis are H plus ions, electrons and oxygen. Okay, I hope that is clear to all of you. Moving on to the next question. During iodine test, the leaf is boiled in methylated spirit to kill the cells of leaf, soften the leaf, remove chlorophyll from leaf, destarch the leaf. Which is the correct answer? What do we do to kill the cells of the leaf? We boil it. We boil the leaf to kill the cells. Okay. Now, to remove chlorophyll from the leaf, what do? We boil in methylated spirit. Here, we boil in methylated spirit to remove chlorophyll from the leaf. Okay. So, first initially after conducting the experiment, we remove the experimental leaf and we dip the leaf in boiling water, and this is done to kill the cells of the leaf. Then the second step in the iodine test is to remove chlorophyll. This is done by boiling the leaf in methylated spirit. Okay, and after that the third step is the leaf is made soft. Okay, when we boil it in a methylated spirit, the leaf becomes hard and brittle, and the leaf is made soft by placing it in hot water. So the third step is making the leaf soft by placing the leaf in hot water. And then the final step in iodine test is to pour a few drops of iodine. And so the leaves or the parts of the leaves which have starch in them turn blue-black in color. Okay, so during iodine test, the leaf is boiled in methylated spirit. Why? To remove chlorophyll from leaf. Option C is the correct answer. Remove chlorophyll from leaf. Question 21. First step in photosynthesis. What is the first step in photosynthesis? Options are photolysis of water, activation of chlorophyll, absorption of a photon of light energy by chlorophyll, and synthesis of ATP. Which is the correct answer? What is the first step in photosynthesis? Photosynthesis comprises of two phases, the light dependent phase and the light independent phase. Okay, in the light dependent phase, that is the light reaction or the photochemical reaction, what is the first step in photosynthesis? If such a question comes, what is it? The first step in photosynthesis is the absorption of a photon of light energy by the chlorophyll pigment. As a result of this absorption of light energy by the chlorophyll pigment, it gets activated. So the first step in photosynthesis is the absorption of a photon. Light energy is absorbed by the chlorophyll molecule, and the chlorophyll molecule gets excited to a higher energy level. Okay, so that is the first step in photosynthesis. The correct answer is absorption of a photon of light energy by chlorophyll. Option C is the correct answer. Moving on to the next question, the process by which several molecules of glucose are transformed into a molecule of starch. So the product of photosynthesis is glucose. Okay, food is synthesized in the leaves in the form of glucose. And this glucose is a monosaccharide and it cannot be stored. For storage purpose, this glucose has to be converted into insoluble starch. Okay, so what is actually starch? Starch is actually a polysaccharide of glucose. Several glucose molecules are linked together or joined together to form a bigger molecule, which is the starch. So what is this process by which several glucose molecules get linked together to form a molecule of starch? Look at the options: photosynthesis, polymerization, photophosphorylation and phosphorylation. We know what is photosynthesis, there is no doubt in that. Now, what is polymerization and phosphorylation? Phosphorylation is the addition process by which phosphate group is added on So here if we take in photosynthesis, we know ATP is produced from ADP, adenosine diphosphate, gets converted to adenosine triphosphate by the addition of a phosphate group. When a phosphate group is added on to ADP, it becomes ATP, adenosine triphosphate. This is a phosphorylation reaction. Okay, when this reaction occurs in presence of light, okay, here in photosynthesis, the formation of ATP is linked with light energy. There is the presence of light energy there, so that is why it is called as photophosphorylation. Okay, since light energy is used in the synthesis of ATP, it is called photophosphorylation. Now, what is polymerization? Yes, polymerization is the process by which starch molecule is synthesized from several molecules of glucose. Okay. So, the correct answer is option B, polymerization. Next question, which is an adaptation in leaf for photosynthesis? Options are thin and broad leaves, presence of stomata, transparent upper epidermis, all the above. Which is an adaptation in leaf for photosynthesis? Thin and broad leaves. Yes, that is truly an adaptation for photosynthesis. Okay, thin and broad leaves. Then presence of stomata. Yes, presence of stomata. What does the stomata do? The stomata, it is through the stomata exchange, gaseous exchange take place. The carbon dioxide that is needed for photosynthesis enters into the leaf through the open stomata. So, presence of stomata is important. Then transparent upper epidermis. Yes, the epidermal cells are transparent. Why? This is to allow light to enter freely into the leaves, because the chlorophyll or the chloroplast are mostly concentrated in the palisade layer. Okay, so for the light to enter into the leaves, the epidermis should be transparent. So the transparent epidermis is to allow more amount of light to enter into the leaves. And the thin and broad leaves, what is the peculiarity of broad leaves? Broad leaves allow maximum light to be absorbed. It provides maximum surface area for the absorption of light energy. And what about thin leaves? The thin leaves reduces the distance between the cells. The distance between the cells for easy transport or fast transport of substances is possible when the leaves are thin. So, thin leaves reduces the distance between the cells to promote easy transport of substances. So, all these options are adaptations in the leaves for photosynthesis. Thin and broad leaves, presence of stomata, transparent upper epidermis. So, the correct answer is option D, all the above. Moving on to the next question. Optimum temperature for photosynthesis. What is meant by optimum temperature? Optimum temperature is the temperature that is best suited for photosynthesis. Okay, photosynthesis is maximum at the optimum temperature. So, the options are 20 degrees Celsius, 35 degrees Celsius, 0 degrees Celsius and 60 degrees Celsius. So, here the correct answer is option B, 35 degrees Celsius. Okay. The optimum temperature for photosynthesis is mostly 35 to 40 degrees Celsius. When the temperature goes above 40 degrees Celsius, naturally it will affect the rate of photosynthesis, because at higher temperature, when it goes more above 40 to a upper temperature limit, naturally the enzymes that are needed for photosynthesis will get destroyed or denatured, resulting in a decreased rate of photosynthesis. Now, if the temperature goes below, that is 0 degrees Celsius, to the lower limit, what happens is that 10 degrees Celsius or less than 10 degrees Celsius, what happens is that the rate of photosynthesis will again decrease very much, because here the enzymes become inactive. There are various enzymes that are needed for photosynthesis. These enzymes get inactive at low temperatures, thereby reducing the rate of photosynthesis or lowering the rate of photosynthesis. So, the maximum photosynthesis occurs at the optimum temperature, which is about 35 degree Celsius. So, the correct answer is option B, 35 degree Celsius. Next question, a plant that is used in the experiment to demonstrate that oxygen is evolved during photosynthesis. Okay, an experiment to show that oxygen is evolved during photosynthesis. For such an experiment, we normally use aquatic plants. Okay, so example, hydrilla. Okay, hydrilla experiment is the experiment to show or demonstrate that oxygen is evolved during photosynthesis. Now here in the options, hydrilla is not there. Instead, options are Croton, Elodea, garden pea, Coleus. Which is the correct answer? Elodea is the correct answer. Option B. Option B, Elodea is the correct answer. So here, why do we take water plants for this experiment is that if we use water where aquatic plants, we can clearly see the evolution of the oxygen gas as air bubbles, oxygen bubbles being evolved from the cut twigs of the plant. So that is the reason why we use water plants for this experiment. Okay. Now next question, that was a simple question. Moving on to the next question, this too is also a very simple question. Starting point in a food chain is always A, autotroph, producer, green plant, all the above. Okay. If you don't read the question properly, there is a chance for making a mistake there. Starting point of food chain is always a green plant. Your eyes will normally fall on that option, option C, green plant, and you will mark it as the correct answer. You will get it wrong. Okay, look at the other options. Autotroph. What is an autotroph? Green plants are autotrophs. They can synthesize their own food using carbon dioxide and water in presence of sunlight and chlorophyll. Producers, yes, green plants are producers. Why? Because they can produce their own food. So, starting point in a food chain is always what? Option A, B and C. All are correct. Option D, all the above is the correct answer. Which is not true about guard cells? Options are, they are bean-shaped cells. They have thick inner walls and thin outer walls. They contain chloroplast. They have thin inner walls and thick outer walls. Which is the correct answer? Just imagine the shape of the guard cells. How is it modified? We know guard cells are modified epidermal cells. How is it different from the other epidermal cells? The guard cells have bean-shaped structure. They are in dicot plants. In this, the guard cells of dicot plants, dicot leaves, you can see the guard cells are bean-shaped cells or kidney-shaped cells. And another important feature of the guard cells is that they have their inner walls thick. The walls facing the stomatal opening, those are the inner walls of the guard cells. They are thick, whereas the outer walls are thin. This peculiar type of thickening enables the guard cells to, when they swell up, when they become turgid, because the outer walls are thin, the outer walls will bulge out. Whereas the inner walls, since they are thick, they cannot bulge out, but they will curve inwards, thereby opening the stomata. So, they have thick inner walls and thin outer walls. That is also true. They contain chloroplast. Yes, guard cells contain chloroplast. That is the difference between guard cells and the other epidermal cells. Guard cells are modified epidermal cells, and one of the differences is that the guard cells contain chloroplast in them. whereas the other epidermal cells do not contain chloroplasts. They are transparent, so that these epidermal cells allow maximum light to enter freely into the inner layers, the mesophyll layer, especially the palisade layer, which contain more number of chloroplasts. So the guard cells contain chloroplast, that is also correct. And because of the presence of chloroplast, as we have seen in one of the previous question, during daytime photosynthesis will take place in the guard cells, increasing the sugar concentration in the guard cells. Okay, so which is not true about guard cells? Look at the fourth option, option D. They have thin inner walls and thick outer walls. That is wrong. That is the false statement. Option D, that is the correct answer. They have thin inner walls and thick outer walls. This is not true about guard cells. Transportation of food through the phloem. What is that process of a food transport through the phloem called? Options are ascent of sap, transpiration pull, translocation, photosynthesis. Okay, ascent of sap. What is ascent of sap? Ascent of sap is the transport of water and minerals through the xylem. Upward movement of water and minerals through the xylem is called as ascent of sap. Transpiration pull. What is transpiration pull? That is the pull exerted on the column of xylem as a result of transpiration. A suction pull is exerted on the water column in the xylem, and it is this pull that causes the upward movement or ascent of sap in the xylem vessels. Option C, translocation. Yes, that is the answer here. The process by which food is transported through the phloem, that process is called as translocation. Okay, and photosynthesis you already know. So what is the correct answer here? Option C, translocation. Transportation of food through the phloem is called translocation. The black paper experiment is used to show that, do you remember that experiment of photosynthesis on which the selected leaf, onto the selected leaf a black paper is placed and the plant is kept in sunlight. After a few hours we do all those experiments, or iodine test is conducted on the experimental leaf. Do you remember that experiment? That is the black paper experiment. When do we use black paper to cover the leaf? Look at the options. Carbon dioxide is necessary for photosynthesis. Sunlight is necessary for photosynthesis. Oxygen is produced during photosynthesis. Chlorophyll is necessary for photosynthesis. Now look at, just think of those different experiments that we have studied. Carbon dioxide is necessary for photosynthesis. For that experiment, what is that we use extra? That is potassium hydroxide, which absorbs carbon dioxide. So the part of the leaf that is introduced in the conical flask containing potassium hydroxide, because of the absence of carbon dioxide inside the conical flask, no starch will be synthesized in that part of the leaf, while the part of the leaf that is exposed or that is seen that is present outside the conical flask will produce starch, because carbon dioxide is available in the atmosphere. So that is carbon dioxide, the experiment to show that carbon dioxide is necessary for photosynthesis. Now let us move on to option C. Oxygen is produced during photosynthesis. That is the hydrilla experiment. In this experiment, we don't normally in all the experiments on photosynthesis, the first step that we do is destarching, isn't it? We place the plant in a dark room for 24 to 48 hours to make the leaves free of starch. In this experiment, that is to prove that oxygen is produced during photosynthesis, we don't destarch. No destarching is done there, because no iodine test or starch presence of starch is not being tested there. So there is no need of destarching the leaves of the plant taken for that experiment. Here the experiment is to show that oxygen, bubbles of oxygen are being evolved during the experiment. This shows that oxygen is produced during photosynthesis. Now, option D, chlorophyll is necessary for photosynthesis. Remember, in this experiment, to show that chlorophyll is necessary for photosynthesis, we take plants with variegated leaves. What are variegated leaves? Leaves having green and non-green parts. For example, money plant, Croton, Coleus, etc. are all plants having variegated leaves. So, such plants are used for this experiment to show chlorophyll is necessary for photosynthesis. So, in such cases, the green colored parts of the leaves will turn blue-black after the iodine test, while the non-green parts do not turn blue-black because starch has not been synthesized in those regions. Now come to the option B, sunlight is necessary for photosynthesis. Yes, the black paper experiment is used to show that sunlight is necessary for photosynthesis. We select a leaf of a plant and we place a black paper onto the leaf. Okay. And what is the conclusion? What do we observe after the experiment? What is the conclusion that we get? The part of the leaf that was covered with black paper do not turn blue-black after iodine test. That is because this region was not exposed to sunlight. Whereas the other regions or the other parts of the leaf which were exposed to sunlight will turn blue-black because photosynthesis has taken place in those regions. So, this experiment, the black paper experiment is used to show that which is the correct answer. Option B, sunlight is necessary for photosynthesis. Next question, the form in which food is manufactured in leaves. Yes, food is manufactured in the form of, look at the options, sucrose, starch, glucose, maltose. The form in which food is manufactured in leaves, that is glucose. Okay, food is manufactured in the leaves in the form of glucose. They are transported from the leaves to the different parts of the plant, mostly as sucrose. Okay, and they are stored in the plant in the form of starch. Okay, so the form in which food is manufactured in the leaves is option C, glucose. Okay, so with that we have come to the end of this session. I hope this session is clear to all of you. If you have any doubts regarding any of the questions, please ask your doubts and give your suggestions in the comment section. Thank you.